Good morning, everyone. Again, I'm Shunichiro Tomura from the University of Queensland. I'm doing a PhD with Mark Cooper and Owen Powell. And today I'm going to introduce about my current research progress, which is about investigation of ensemble approach for gene prediction in crop breeding. And then in the last year's symposium, I demonstrated that ensemble approach was able to increase the prediction performance for gene prediction. But this year, I'd like to go one step further by investigating why ensemble was able to increase the prediction performance in the gene prediction in crop breeding. But before I start to explain that in detail, let me quickly introduce about why I'm doing this and my research targets so that we can understand my research goal for my PhD. And my target for my research target is a selection accuracy increase in this breeder's equation. And by doing so, we can increase the selection. This one here, right? So that we can increase the gene gain as a result. So that's my goal for my PhD. And then one approach to achieve this goal is the application of the one method called the ensemble approach. So in this case, we build a number of different gene prediction models. And then they can collaborate. And then by the end, by aggregating all the information coming from each of the single models, we can further expect to increase the prediction performance by refining the final prediction. So that's the conceptual understanding of the ensemble approach. So we can now start discussing about our experimental design and then the result and so on. So in our case studies, we use 3 different maize datasets. The first one is a genome dataset from Chen et al. 2019, which is about the crosses between the 1 common parents from maize W22 and then the 5 different genotypes from teosinte, which is the ancestry species of the maize as a dominant parents. This generated 5 different populations. And I also used this dataset for the last year's symposium, but like I said, we have 2 more datasets here to make more generalized. The second one is maize landrace dataset from Mayer et al. 2020. This is quite different from the previous one. This is because we have 3 different genotypes coming from the wild maize types, and which is crossed with maize coming from 65 breeding lines from the diversity panel. So that's quite different from the previous one. And then the last one is much more difficult again, much more different again, which is a maize dataset from Buckler et al. 2009. So in this case, we have 2 parents again, common parents, maize B73, and then the 25 different cultivars coming from both temperate and tropical regions as a dominant parents, generating 25 different populations dataset. And we chose those 3 datasets because we can observe the diversity in their gene marker. So for example, if you think about genome dataset, their gene marker is much more diverse because of the teosinte. So the condition is much closer to the wild condition in terms of their gene marker, whereas the maize dataset here is much less genetically diverse because of the domestication process in the past. So that's why we chose 3 different datasets, and we kind of expect to have some kind of effect towards the performance of gene prediction by the ensemble. So this is the actual ensemble workflow in this experiment. So first of all, we developed 6 different individual prediction models, GBAP, base B, RKHs, random forest, self-vector aggregation, and multi-layer perceptron. And then we train and evaluate those 6 different models by using training and test datasets. And after doing that, we get the predictive phenotypes from each of the single models for the test dataset, and then they'll be aggregated into one to make the prediction matrix, which will be used as an input for the ensemble approach. And then in terms of algorithm, we use one simple approach called naïve averaging method, which simply takes all the prediction result from each of the single models and then average them into one with the same weight. And again, we have to evaluate its performance. And in terms of evaluation, we use one metric, Pearson correlation. And then the target traits are all related to flowering time, the antithesis, the to silk, and the antithesis silk interval. And then this is actual prediction result for the both single models and ensemble approaches. And for the 3 different datasets across the 3 different traits. The blue boxes are representing the average performance of the single models, whereas the orange boxes are representing these ones, representing the performance of the ensemble approach. Then, as you can see, that the box position of the ensemble approach is always higher than the location of the single model's average, indicating that this ensemble approach was able to increase the prediction performance and outperform the single models average in all dataset, which can lead to the increase in selection accuracy and the gene gain as a result. And one interesting observation here is that the gap between two boxes are different by, this gap size is different by the dataset. For example, if you think about telenom dataset, the gap between the two is quite large, but such large gap is not observed for the other rest of the dataset here. So that's it can be one interesting research question. So this is like this. And why is there a difference in the gap size differences between the two boxes? And one explanation can be the Powell ensemble, depends on the level of the information diversity obtained from the single models. So this is because when we ensemble each of the single models into one, and then each of the unique information can be comprehensively captured to make a better prediction performance. But if each of the single models capture a similar information, then the ensemble just generated another similar prediction information, then which won't be able to increase the prediction performance that significantly. So, but this explanation can be reasonable, but we have to understand whether this is true or not, or at least we can understand. We have to validate whether this understanding were right or not by constructing one plot called a scatter plot matrix like this. So this is for the data and thesis for telenom dataset as an example. So this one does the pairwise comparison of each of the single models using this experiment. The top three are coming from the classical conventional prediction models, and then the last three are coming from machine learning models. Yeah, and then as you can see, we have two different main parts here. The top part is the first part, representing the right triangle in the green. This shows the pairwise comparison of the models at the predicted phenotypes, whereas the bottom part is doing the pairwise comparison of each of the single models at the level of the marker effect. I know this is very complicated, so we can say many things from this scatter matrix, but one observation can be that we can see some positive associations in the both triangles. So for example, if you think about GBAP and the base B at the level of the predicted phenotypes here, we can see a linear trend like this, right? Here, and at the level of the marker effect also, it's weaker, but we can see some kind of like a trend like this here. So, but that's one message. But the one more significant message here is that we cannot see such a linear trend by having a little associations in many pairs. So, if you think about the multi-layer perception and then the other 5 prediction models here, you can see such a clear linear trend by having a clusters at predicted phenotypes. And also, we can say the same thing for the RKHs and against the 5 different single models at the level of the marker effect level here. So, but like I said before, this diversity in information can be the key component that can enhance the prediction performance by the ensemble. That's the one key message from the talk. And now we can construct the same scatter plot matrix for the maize number dataset. And it's obvious, I mean, in terms of the tendency. We can say that we can see much more clear linear trend here for the maize dataset for predicted phenotype level and some parts on the marker effect level as well. This indicates that we can see less diversity in maize dataset in terms of their captured information. And then this diversity information in the maize dataset might have reduced the advantage of the ensemble as or a hypothesis. And then we were able to see the same trend for the maize long race as dataset as well, indicating that this diversity in the information can be a key factor contributing to the prediction performance enhancement by the ensemble. So that's one message here. But now, since we can extract all the information from each of the single models in the SNP marker effect here, so we can go one step further by doing the comparison of the estimated marker effect with the prior knowledge by constructing another plot called the circos plot. I'd like to do this because by doing so, we can check whether predictions are based on the actual key gene regions and interactions. And this is one example of the circos plot for the for the data and thesis across populations telenom dataset. And this is very complicated. So I'd like to explain each of the components one by one by separating it into 3 different ring components and links, so that we can understand what's going on. Yeah. And for the first component, it's coming from the QTL ring, representing the QTL regions detected in China 2019. Because this, again, this is a telenom dataset, so I just use the QTL information from the Chantal's research. Like this is the most inner part of the ring area, ring section. And then this represents the one prior knowledge of the QTL regions and key gene regions. And the other component representing the prior knowledge is the gene region rings based on the information from the Dong et al. 2012. They classified a number of different key gene regions, and then they classified them into 7 different roles based on the functions. And then based on the information, they developed a network here, gene network affecting the data entities for maize. And then based on the information, we also developed the 7 different rings. Each of them is corresponding to the one of the 7 rows here. And also put the 3 different colors to it. So if a gene region affects a leaf, should apical meristem stem or both leaf and stem, the color will be the green, the orange, and purple respectively. So that's the second major component showing the prior knowledge. Now we can compare those 2 different components against the estimated marker effect coming from each of the sync models by doing a back calculation method in this experiment here, right? And then we can see the darkness of the color is different. So if it becomes darker, then it means the marker effect becomes stronger and stronger. But one interesting observation here is that although each of the sync models has its own unique mechanisms in terms of their prediction, but their overall tendency is kind of similar by highlighting a similar part as like a highly important like this. But however, if you focus on the granularity of each of the highlight region, for example, this part, and this part and this part, for example, we cannot see like some consistency. We can see some like a bit of like a variation in terms of the darkness. That's a connection between the previous scatterplot matrix by having a little one associations. That's about one of the interesting message. So now we can move to the link part representing the marker interactions estimated by random forest using the one machine learning interpretable method called Shapley values. And thickness of the line shows the strength of the interaction. And stronger means thicker means stronger, like this part here. And then we can see red and blue, they're represented within chromosome. This one, yeah, here, like this, and between chromosome, like all over the place, like this. Yeah. So that's all the components from the circus plot. So now we can assemble all the components into one and compare that against the network from the Dong et al. 2012 here. Yeah, their network is complicated as well, but the one key message here is that we can see many of the regions from chromosome 8, 1, and 10 are highlighted, and they were highly interactive with each other. But if you come back to the overall circus plot here, we can see that many regions, like several regions, chromosome 8, 10, and 1 are highlighted as important. And in those regions, some of the parts are highlighted with some like prior knowledge components like this. Yeah. So that's the interesting part. And also we can see some interactions, heavy interactions within the 3 different chromosomes within the highlighted regions here. For example, if you think about this ZMCCT, it's highly interactive with the chromosome 8 here. And such interaction at this part, its dark region is a ZMCCT from the prior knowledge. This has a heavy interaction with the chromosome 8. This part is a ZCN8 here. That's why we can say that each of the single modules are capturing heavy interactions between the chromosome 8, 1, and 10. And also we can develop the circuit plot for all the dataset, and if we observe them, some of the parts are conserved, representing those regions like interests and regions and interactions for the maize. And also we can see different parts. This shows this difference in pathways is showing because of the domestication process in the past. And we can see the many different components by the circuit plot by the population for 10 of the dataset. We can see diversity in the key gene regions interactions. They're showing that the difficulty in population prediction because their key characteristics are different by the population. So we can say that ensemble approach was able to capture the prediction performance and enhance its prediction ability. And then the width and the strength is coming from the information diversity. And then the prediction is actually based on the key gene instructions and the information coming from each of the single models. And then we have 3 future outlook and plans. But I won't go through each of them because of the time. But by think about those three different components, we can further enhance the prediction selection accuracy and then gain as a result, which is my research target for my PhD. That's the end of my presentation. Thank you for listening.
Thanks a lot. Very nice. One question.
Yeah, thank you for your presentation and interesting research. I was wondering, I mean, at the beginning you showed the comparison between the ensemble of the 6 models as opposed to just the average of these two, of all these 6 models. I was wondering, did you see differences, clear differences in performance of the individual models?
Yeah, it's not clear. I mean, yeah, I cannot say it depends on the data set, but in many cases, they're not, it's not showing the clear like a difference in performance. They're kind of average, but yeah, that's why I'm saying it's kind of a limitation of each of the same models. That's why I came up with this ensemble approach to enhance the prediction performance. Yeah. So the answer is like we couldn't see the clear like difference gap in performance.
So you did not have clear ideas on whether to leave these models in the ensemble or leave it out together.
Yeah. So that's my feature.
Scatter plot matrices showed the difference between the models. Some were highly correlated, others were not correlated at all.
Yeah, among each other, but then, yeah. Okay.
You'll catch up later.